Hi everyone. Today we start with the next chapter, which is cell cycle and cell division. We have just completed the cell chapter and I hope it is all clear and very much fresh in your mind. So we ended that chapter by discussing nucleus, right? In the nucleus we discussed all the different parts of nucleus, and finally we also saw different kinds of chromosomes, right? Keeping that in mind, from today we will be beginning with cell cycle and cell division, right? Now you may be wondering what is the need for cell division, or if you already know, let me revise it for you, okay? So basically, let us consider this small baby, okay? This baby has to grow up, okay? It will grow up, she will grow up. She will go to the primary school, then higher secondary, she will go to the college, become a working woman, and then become an old lady, right? So there is this growth that you see. And in this growth, which is both mental, physical, and in every manner, all of that growth is only possible if there is cell division, right? So basically all the different kinds of organism, they start with a single cell, which is called a zygote, right? Zygote is after fertilization of egg or ovum with the sperm, there is a single cell found which is called zygote. This zygote will undergo multiple divisions and finally lead to formation of a new baby, right? This is the case even true for plants. So this is the first need for cell division, that you need to grow, right? Second thing is, for some protista, okay, for some unicellular organism, cell division is also a manner of reproduction, okay, because these are unicellular organisms, they do not have many cells, they do not have a whole body. So what do? If they undergo division, it will lead to formation of two new organisms, which means it is a way of reproduction, okay, to make more and more of its own kind. And the third thing is, do you remember when you were driving a cycle or when you fell down and there were a few scratches, but those scratches heal, right? So this healing process also occurs because of cell division. Just look at this diagram. There is some skin wound, okay? Wound, there is some scar, and some cells have died. They have just come off, right? So there should be other cells which will divide and fill up this gap, okay? And as they fill up this gap, your skin or your tissue, it becomes normal, okay? There is proper healing. So these are the you should keep in mind why the cell division, it is important, okay, the basics. Okay, so you know what cell division is. Division is breaking down something into two parts or more, right? Now what is cell cycle? As the name suggests, it is some cycle which goes on inside the cell. So cell cycle refers to a series of events. It is not just one thing, it is series of events, okay, a lot of things happening one after the other that takes place in a cell. What does it result into? What does this series of events do? It results into duplication of DNA, so your DNA becomes double, okay, it duplicates, and division of cytoplasm as well as organelles to produce two daughter cells. Fine. Just imagine, let us say I have some notes, okay? I have a book and I want to give it to two of my students, but I just have only one book, right? Same is the case with your cell. It has only one nucleus and one DNA. So how will it give it to two daughter cells? So the first thing it has to do is duplicate its DNA. So that is what happens in a cell cycle, duplication of DNA. So I will do what? I will get a Xerox copy done of that book. In the next stage, I will give one book and one Xerox to the students, right? So that is the whole cycle is what is called cell cycle. Clear? It is all the preparation a cell does to divide its own material into two parts, okay, into two daughter cells. It is mostly divided in different phases, as we shall see, and you will see it in this cyclic form, which means that it keeps on repeating itself, right? So it is always in a cyclic form, never in a linear form. The cell cycle was discovered by Prevost and Dumas. And they were studying the zygote of frog. Okay, while studying the zygote of frog, they came to know about cell cycle. Now, time taken for this cell cycle varies from organism to organism and cell to cell. Okay, how much time will a cell take, or how much time will an organism take to duplicate this DNA and to get ready before division? How much time will I take before I can go to the stationery shop? I can give the book, that stationery shopkeeper, he would, let us say, Xerox, okay, photocopy the whole book, and then I would come to you and I would give it to the students. So this whole process, this whole cycle, right, it takes time. And this time taken, it varies from cell type to cell type. Okay, it may be different for your cells in your eyes, or cells on your skin, or cell in your stomach, right? And it is also different from one organism to another. The time taken for your cell to divide may not be same as time taken for a lion's cell to divide. Fine. Let us see it with an example. Yeast cells, they divide in 90 minutes. Okay, in 90 minutes a yeast cell will divide into two. Right, here you can see these are the yeast cells and they are dividing. Can you see here a bud coming off, and whoop, now you have two cells. So for a yeast cell, which is a unicellular eukaryotic cell, a protist, it takes about 90 minutes before it can divide. Okay, a human cell can take up to 24 hours or even more. Okay, so for humans it takes 24 hours to for a cell to divide and become two. For bacteria, okay, this is for a human cell, okay, especially in case of a zygote, you will see there will be this one cell, okay, it will start getting divided into two. Just wait for it, it takes 24 hours. See, now you have two cells. Fine. Bacteria, like E. coli, it can divide in 20 minutes. That is, every 20 minutes you have double the bacteria. Every 20 minutes, double the bacteria. Okay. So this shows that how variable this cell cycle can be. A typical eukaryotic cell cycle is divided into two main phases. Now, whatever we shall be discussing in the chapter are on the basis of, we will consider only eukaryotic cell. Fine. The eukaryotic cell cycle is divided into interphase and M phase. Interphase is the preparation before the cell divides. Okay, all the preparation I do, I go to the shop, I get the book photocopied, and then I come to you, right? So all of this, that is interphase. And then you have M phase. M phase is mitosis or meiosis. It could be either or. We shall study both of these one by one. So here is the course division of your cell cycle. The cell cycle can be divided into, as we just saw, interphase and M phase. Interphase itself is divided into three phases, okay, or three parts: G1, S, and G2. M phase is also divided into two parts. The first part is called karyokinesis. Always remember, you have heard this word a lot of times, karyo, right? You have heard it in prokaryote, you have heard it in eukaryote. So what is this karyo? Wherever you hear this word karyo linked to any of the word, it means something related to nucleus. So what is happening here? There is nucleus dividing. Okay, so division of nucleus is called karyokinesis. And then there is division of the cytoplasm and all of its contents, which is called cytokinesis. Karyokinesis, which is the division of nucleus, is again divided into four parts: prophase, metaphase, anaphase, and telophase. Clear? Do not worry, we will go through all of these one by one. Let us start with interphase, and in that G1. Interphase is the resting phase of the cell cycle. Okay, here the cell is at rest. Actually, it is not really at rest, because it is undergoing a lot of metabolism and growth. But because when you observe a cell which is at interphase under a microscope, you will not see a lot of activity, which is why people thought it is a resting phase, that the cell is doing nothing, while in fact the cell is doing a lot of metabolism and it is doing all the preparations which are not visible to you. Okay, so cell prepares for division by cell growth and DNA replication. Fine. Just think about it. If a cell, it has to divide, okay, it has to give all of its components into two daughter cells equally, then of course it has to grow in size, right? It has to grow big, it has to produce a lot of organelles, a lot of nutrients, a lot of DNA. So this means it has to grow before it can divide. Okay, always remember this. If it does not grow before it divides, then the cell size will keep on reducing over time, right? That does not happen because during interphase the cell grows. The cell organelles divide and there is increase in the cytoplasmic content. There is no visible change under the microscope. Okay, visibly when you look at the cell under a microscope, you will not see any change. It takes about 95% of the time of cell cycle. Okay, if this is your cell cycle as shown here, then your interphase, it takes about 95% of time. Okay, apart from this pink colored region that you see in this pie chart, the whole circle is covered by your interphase. Clear? It is divided into three phases. You have the first phase, which is called G1, where you can see there will be cell growth. Then the second phase is S phase or synthesis phase. This is the phase where there will be DNA synthesized. Okay, this is the phase where I will go and get the book photocopied, right? And after that, there will be preparation for cell division. There will be some growth which is left, which will occur, and then the cell will divide. That actual division is here, okay, in mitosis, okay, M phase, and then you have cytokinesis where the cytoplasm divides. Chalo, let us start with the G1 phase. G1 phase is also known as gap 1. Okay, the name comes from gap 1, or you can also call it growth phase. Okay, that is why you always remember G1 is for growth. This is the phase between mitosis and initiation of replication of DNA. So, some mother cell might have divided and given rise to this daughter cell. Now, before this daughter cell itself can go and divide, it needs to grow, right? So, this phase, this interphase, is between mitosis, okay, one mitosis, and the DNA synthesis, okay, this G1 phase. Here, the cell is metabolically active. It is producing a lot of ATP, okay, it will be utilizing a lot of nutrients and producing a lot of ATP, fine, and this ATP will be used for anabolic reactions, that is growth, okay, it will be used for growth. Cell is at its maximum possible size, okay, the cell will increase to its maximum possible size in G1 phase, fine. Here, there is increase in synthesis of RNA, all types of RNA. It could be mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, that is ribosomal RNA. All of these RNAs will increase. Then, because mRNA is getting, the number of mRNA are increasing, the enzyme and protein production will also increase. Fine, so you will get more and more protein, more and more enzymes. What are these enzymes and proteins required for? So they are required for DNA replication. Always remember this as a thumb rule. Whichever phase we will be discussing, for example, we are discussing G1 phase, then the activities taking place in this G1 phase will be to help the cell perform function in S phase. Okay, so whatever we saw in G1 phase, is just preparation for the S phase. It is like how you are preparing yourself every day. You are reading every day, you are solving questions every day. For what? You are waiting for NEET 2021, right? So here, G1 phase, you are just preparing for the S phase. In the S phase, you are preparing for the mitosis, right? So in this way, just a general thumb rule that in whatever phase you want to describe or discuss, you are just preparing for the next phase, okay? Helping the cell function in the next phase. So now we move on to the second phase of interphase, which is S phase. In S phase, which is synthesis phase, DNA replication takes place. I hope you remember from your molecular biology chapters, okay, that this will be one strand of DNA. This one strand of DNA will be separated, okay, it will give rise to this replication fork. Then in this replication fork, DNA polymerase, which is an enzyme, this DNA polymerase was synthesized in G1 phase, right? But it is functioning in it is functioning in S phase. So here that DNA polymerase will start synthesizing new strands of DNA. Clear? These new strands of DNA finally will now give you two same types of DNA molecules, right? So this is what is DNA replication or DNA duplication. Fine? Here, there are some important things you need to remember. Fine? So we will go through it slowly. Just keep up with me. Fine? If the initial quantity of DNA, which is this one strand, if this initial quantity of DNA is 2n, then after replication it becomes 4n. Clear? Now let me show you why it would be 2n. In a diploid organism, okay, diploid meaning they have a pair of chromosomes, one from each parent, right? So even for you, you have 23 pair of chromosomes, which means for you write diploid organism as 2n, right? You have a pair of homologous chromosomes. So this is written as 2n. And the amount of DNA here is 2 capital N. Fine. Here what is shown is one chromosome and this is one single chromatid. After replication, the diploid organism is still diploid. It is not becoming tetraploid. So 2n will remain 2n. Okay, do not make a mistake here. 2n, the diploid organism, is still a diploid organism. Fine. But the amount of DNA has doubled. Clear? So at first you had one chromosome, one chromatid. Now you have, this is still one chromosome. Okay, this whole structure will be called one chromosome, but it now has two chromatids. Okay, and these are called sister chromatids. Why they are called sister chromatids? Because they are same. Okay, they have the same DNA. Fine. So the amount of DNA will go from 2 capital N to 4 capital N. Clear? But the diploid organism, which is 2n, will still remain diploid. It is not becoming tetraploid. Fine? The number of chromosome does not change. Very, very, very important. You will still have 23 pair of chromosomes, but these 23 pair of chromosomes would have now become divalent. That is, they would have two chromatids. Clear? The centrioles in the centrosome also divide into two. So here there would be in the cell, let me show you. This is one centrosome. We have already studied centrosome, in which there are two centrioles arranged at right angle to one another. The number of centriole also becomes double. Okay, so here the whole centrosome or the two centrioles, they divide and form a whole new centrosome. Clear? So now in S phase, synthesis phase, what you have to remember, your chromosome has gotten one more chromatid. Okay, so it has gone from one chromatid to two. The centrosome has divided into two, and also there will be synthesis of histone proteins. Why are these histone proteins synthesized? Because this diagram, what I had just shown you, is just for reference. Okay, in interphase you do not really see chromosomes condensed like this. The condensation will happen now. Fine, this was just to make you understand about this 2n and 4n, but till now your DNA is still in this form, okay, in this fibrous form. So histone proteins are synthesized in the S phase, and these histone proteins, as we had discussed last time, will help in packaging of the DNA. Okay, it will help in condensation of the chromatin to make chromosomes. Now after S phase comes the GAP2 or G2 phase. This is the final preparation before mitosis. You are preparing the cell for mitosis. Okay, here you can see this is your nucleus. The nucleus still has chromatin. It will also have histone proteins, okay, plus DNA, which is chromatin. The centrosome has divided into two, so you now have a pair. The RNA, proteins and other macromolecules required for multiplication are produced. Whatever RNA, proteins and other macromolecules are needed during this multiplication process are produced here. Especially in this proteins, what I would like to tell you is about tubulin protein. Remember the microtubules, right? They were a component of your cell cytoskeleton, fine? So these microtubules will also help us in the cell division process, okay, in separation of the sister chromatids. Here, this tubulin protein is synthesized for formation of spindle fibers. Okay, these spindle fibers are nothing but they are microtubules. But the fibers which are helping us in cell division are just specially named as spindle fibers. Clear till here? We have seen growth 1 or gap 1 phase, G1. Then S phase where the DNA was synthesized, histone was synthesized, and centrioles divided. And then we saw G2 phase where the cell started producing tubulin protein, okay, and it is now ready to undergo mitosis. Before talking about mitosis, there is one more phase in interphase seen only in some cells, which is called G0 phase. So here this is your cell cycle. The cell is growing, it is dividing, and then the cycle keeps on continuing. G0 is cell arrest. Okay, the cell will stop. It will come out of this cycle. It will not continue the cycle of division. This is called G0 phase or cell arrest or quiescent phase. How to remember quiescent is just remember quiet. Okay, the cell is quiet. It is not doing anything. So G0 phase is cell arrest under nutrition deficiency or some stress. When there is not enough nutrition, then you should not do cell division, right? So under nutrition deficiency or if there is some stress, there is stress of dehydration or there is some other stress like temperature, then you should not really divide at that time. So the cell gets a signal that you should come out of this cell cycle and stop dividing. This is called the G0 phase. Here, the cells are metabolically active. They are alive, but they do not divide, okay? Some cells do not divide at all once formed or differentiated. Fine? Differentiated is when a normal cell, it divides and it takes up some special function. For example, there was a common cell which divided and now it has taken up the function of heart muscle, right? So we can call it a differentiated cell. It is knowing what it is doing, right? Then RBCs can be called differentiated cells, right? So these differentiated cells, once they are formed, they do not divide, like heart cells, like your neurons. Some divide only when replacement is required for injured or dead cell, right? Especially on your skin. If there are some scratches or if there is a wound, only then will the cells divide. Otherwise, they stay normal. Okay. These cells, when it is required, they exit the G1 phase and enter this G0 phase. Now, I would like to just go through this point briefly. Then there are some checkpoints. Okay, the cell cycle cannot go on and on without any checkpoints. Checkpoints are, you must have seen where the police stand and they just check all the vehicles and they make sure everything is all right before the vehicle is allowed to continue its journey. Right? Same thing happens here. There is a checkpoint in G1 phase, okay, G1S phase. This is called cell growth checkpoint. Here, basically, the cell will check whether it has grown enough in size, if it has enough nutrients, if the conditions are okay. Okay, if there is no stress, there is no nutrition deficiency. Only if all these things are okay, then the cell cycle will continue to the S phase. If there is any problem, if the cell is not the right size, or there is not enough nutrition, or any other reason, the cell cycle will stop here and the cell will go into G0 phase, which is resting phase or cell arrest. Once that checkpoint is crossed, that yes, the cell is ready, everybody is ready, everything is fine, let us do DNA replication now. Then during this S phase, there is a DNA synthesis checkpoint. What this DNA synthesis checkpoint does is it checks the DNA. Okay, so what I will do after going to the stationary person, he will photocopy the book, and now I will just check whether the photocopies are exactly the same or not. I will check if there is any page which has gone missing, or is there any misprint or improper print. So I will do this checking right before giving it to the students. Same thing, the cell is checking whether the this DNA replication has occurred properly. If there are any mutation or if there is any part of DNA which is missing, then the cell cycle can stop here as well. Okay, the cell will stop. It will say repair your DNA and then go ahead. Fine. If it is repairable, then good. If it is not repairable, if it cannot be repaired, then the cell will die. Okay, and this death will be programmed cell death or apoptosis that we studied during lysosomes, right? Fine. So now your cell is healthy, it is ready, the DNA has duplicated, it is also okay, both of the copies are fine. Then the cell will go into G2 phase and M phase, mitosis phase. Fine. There is one more checkpoint in mitosis which I shall discuss when we reach that part. Fine. So this was interphase G1, S and G2. This phase is common between mitosis and meiosis. Okay, this is common phase between all different types of cells. There will be meiosis or meiosis, it does not matter. M phase is also called division phase or mitotic phase. In mitosis, the number of chromosomes in the parent and the daughter remain the same. So it is called equational division. Okay, there is equality. I am not giving half book to one student and half book to another student. I am giving the whole book, okay, the complete information I am giving to both of my students. So same way, in mitosis, there is equational division. The parent cell gives equal amount of DNA to both the cells. Okay. It takes about 1 hour in the 24-hour cell cycle of human. Okay, so when I said that human cell cycle, that is from one cell to become two, it takes 24 hours. So out of that 24 hours, only 1 hour is taken for this mitosis. In animals, mitosis is seen only in diploid somatic cells. Okay, only in diploid somatic. Somatic matlab all the cells except your gonads. Okay, all the cells of the body except your gonad cells or the cells which produce gametes. Okay, so any cell of your body which is diploid and somatic cell will show mitosis, except in some insects. Remember honeybees? Honeybees, you must have seen that though they are diploid, they can give rise to haploids also. right? Same thing with ants. You will study it in genetics, so do not worry. For now, just remember, in animals, almost everyone, okay, every animal, the diploid somatic cell undergoes mitosis. In plants, however, mitosis is seen in both haploid and diploid cells. This again we shall discuss when we talk about plant classification. Okay, there the last portion of the chapter is the life cycle in plants. So do you remember there is haploid gametophyte, which can also grow into a nice tree. Okay, and there is diploid sporophyte, which can also grow into a multicellular um, uh, plant, right? So in plants, mitosis can be seen in both haploid cells as well as diploid cells, while in animals it is seen only in the diploid cells. This phase is very clearly visible through microscope. So if you have a microscope or you are looking through a microscope, M phase will be very easily identifiable. It starts with nuclear division, which is called karyokinesis. Okay, again, this word karyo means nucleus, kinesis means division, and ends with division of cytoplasm called cytokinesis. Let us start with the M phase, the first part of M phase, which is prophase. Okay, pro, wherever you hear the word pro, it means first. Okay, it means good at something. Like you say, this guy is pro. Pro means he's first, or he's in the beginning, okay, of all the maybe sports or whatever you're discussing about, right? So pro phase, so this is a phase which comes first, pro. What happens in prophase? Now just remember, in G1 phase, you are ready with the DNA which has duplicated, and where you are ready with the histone proteins. So in this phase, in prophase, this histone proteins and the chromatin and the DNA, it will start getting wound up, okay, and it will start producing chromosomes. When the chromosomes start to condense, you will notice that it is a bivalent, which means it has two chromatids, fine, sister chromatids. Golgi body, endoplasmic reticulum, nucleolus, and nuclear envelope. This will all start to disappear. Okay, it will all start to degrade a little. The other cell organelles, which are not mentioned here, right? The other cell organelles, they will be attached at the periphery. Okay? They will be at the cell periphery, in the surrounding, not in the center. The center has to be empty. So all the cell organelles which will move towards the side, okay, they will all get accumulated on the sides. The centrioles, which had duplicated during S phase, now start traveling towards the pole. Okay, they will start traveling. One will move towards north and one will move towards south. Now there are two types of mitosis: Estral mitosis. Just let me just tell you before that what ester means. In reality, ester, when you just search for it, it means star. Okay, and here what we are talking about, estral rays or estral microtubules are the tubules or microtubules coming out of the centriole. It makes the centriole look like a star. Okay, that is why they are called esters. So this is what is seen in this is basically what is seen around the centriole, esters, right? In higher plants, centrioles are absent, right? So there are no estral rays which are formed. There will be no such estral rays or esters. Then the other type of mitosis is amphiestral or estral mitosis. In this case, the animals, they have centrioles. Okay, even lower plants like fungi and some algae, they all have centriole, right? So they will also show such kind of esters or estral rays. Clear? Right. So this is how the prophase looks like. The centrioles are moving apart. Then the plasma membrane, nuclear membrane, my mistake. Nuclear membrane is disintegrating, it is dissolving. The DNA is now condensing and forming these kind of chromosome-like structures. If you look at it under a microscope, this is how it will look like. This is the cell for your interphase or resting phase, okay? And this is the cell where the chromatin has started condensing. You can see there is no border, there is no outline, so the nuclear envelope is absent, and the DNA has started condensing, so you can see it thick, okay? At some parts you can even see it to be darker than the other parts, right? So this is the early prophase, and as it keeps on condensing, you start seeing proper chromosomes. So this is late prophase. After prophase comes metaphase. In metaphase, there is complete disintegration of nuclear envelope. There is no nuclear envelope remaining. Okay, the chromosomes, they spread out throughout the whole cytoplasm. Okay, the chromosomes can be present anywhere in the whole cytoplasm. Condensation of chromosome is complete. They are in the most condensed form, the most dense form, and they have two sister chromatids. Okay, this one chromatid and this another chromatid joined together at this black colored centromere. On the centromere, there is this disc shaped thing called kinetochore, right? We have already discussed this kinetochore as well. Here I wanted to show you, this is what it looks like. Okay, this is kinetochore, and this is your microtubule or your spindle fiber. So the spindle fiber will come and it will attach to the green colored kinetochore like this. Okay, so this kinetochore is the site of attachment of the microtubules. Then there are polar microtubules which help push against each other. To understand this better, let me show you this diagram. Here you can see there are three kinds of microtubules. Okay, there are three types of microtubules. First, you have estral microtubules or estral rays. These estral rays will help attach the centrioles to the cell membrane. Okay, it will help attach the centriole to this part of the cell membrane. Okay, opposite poles. Then you have these blue colored ones which are attached to the chromosome. These are called kinetochore microtubules, okay, because they are attached to the kinetochore. And finally, you have third type of microtubules called interpolar or just polar microtubules. These polar microtubules, what they are doing is they are pushing against each other. Okay, it is like have you seen wrestling or kushti or something, right? So in Kushti, think of Salman Khan doing Kushti in Sultan or some other movie, right? So what do you see? You see two wrestlers. They will be pushing against each other, right? And when you push against each other, you are moving away from each other if you keep on pushing, right? So this push is done by interpolar microtubules to separate these two sides. Fine, something should be there to separate this side of the cell and this side of the cell, right, to push against each other. The kinetochore microtubules will pull, okay, like how you pull how you, let us say, how you pull a door, right? So, or pull something, let us say from the well, how you pull the bucket of water. The same way you will be pulling through this kinetochore microtubule. So in this metaphase, now what is happening is you have, through this push and pull, all the chromosomes, okay, they will be aligned at this central plate. This central plate is called equator or metaphase plate. Okay, it is a two-dimensional, what you can say, plane. Okay, and this plane or this plate is called metaphase plate. Here, one chromosome has two kinetochores on each side, and both of these kinetochores will be attached to different sides of the centrioles. Clear? And this is how they will align at the center. In case of humans, okay, in case of our body, we have 46 chromosomes in total. So if you look at a human cell and the metaphase plate, we will have 46 chromosomes aligned at the center. Clear this much? Okay. Right, so this is what equator means. Okay, on a globe you have two poles and you have the middle layer, middle portion, which is called the equator. And this is what will happen. You have this metaphase plate. These chromosomes which are lying around in the cytoplasm will come together and form this straight line. Fine. After metaphase comes anaphase. In anaphase, the centromere, okay, which was holding the two chromatids together, this centromere, it splits lengthwise, fine, in all chromosomes at the same time. So here we were seeing there were different chromosomes, okay, aligned at the same plate. All of these will be splitting lengthwise at the same time. The sister chromatids, they will now separate, and now each of these sister chromatid is a single chromosome. Clear? They used to be sister chromatids when they were attached. Now that they are separated, they are one full chromosome. Okay? So they can be called individual chromosome. At this point of time, the whole cell has two copies. Okay? It has double the number of chromosomes. Clear? So now at this point of time, you will have how many? 46 plus 46 chromosomes. Clear? Each chromosome is pulled towards the opposite pole. Okay, one will be pulled toward the north pole and another one towards the south pole. As the kinetochore microtubules shorten, these kinetochore microtubules will keep on reducing in length, and this is how the chromosomes are pulled apart. Like this. You had all the chromosomes at the metaphase plate, and now they will separate. Clear? Now this separation, I just wanted to mention something because I saw that once or twice such a question was asked. So what is keeping these two sister chromatids joined? There is a protein called cohesin. Okay, remember cohesive forces, adhesive forces from your transport chapter? If you do not, cohesion is basically the attraction, okay, between two molecules. So here the protein is called cohesin because it is attaching this one sister chromatid to the other sister chromatid. Fine, it will be attaching here. Now what happens during anaphase is there is a complex called anaphase promoting complex, APC. The name itself suggests what will it do? It will promote anaphase. This anaphase promoting complex will activate an enzyme called separase. From the name itself, you know what will separase do? It will separate. So here you can see the cohesin protein has been degraded. Okay, you have cut it down. So when the cohesin proteins are cut down, you can easily separate the sister chromatids. And this is how in anaphase, the two sister chromatids are pulled apart. The final phase of karyokinesis or M phase is telophase. In telophase, now the clusters of the chromosomes, okay, will be on different poles. The nuclear membrane will start appearing. Then endoplasmic reticulum and other organelles will also start appearing. The the chromosomes will start to decondense. Okay, and as they decondense, they will get converted into chromatin. So basically now here you have these two beautiful nucleus in one single cell. Now all that is remaining is to divide the cytoplasm. That part is called cytokinesis. The cell is divided into two daughter cells by separation of cytoplasm. The process is called cytokinesis. In animal cells, this is how it happens. This is one cell which has now gotten two nuclei, right? In between, in the middle, exact middle, there will be actin and myosin fibers arranged. Okay, like rubber band. Think of a rubber band. Okay, this is a water balloon and think of a rubber band that you are putting in the middle. This rubber band or this ring is called contractile ring. Okay, and as the contractile ring becomes narrow and narrow, it starts forming something called cleavage furrow. Furrow is this what you can say fissure that you see. Okay, this line that you see, this incision that you see is called cleavage furrow. As the ring keeps on contracting, The cleavage furrow, it moves from outside to the center of the cell and it divides the cell into two daughter cells. Clear? And now you have these two daughter cells. Fine. In plants, you cannot really use a cleavage furrow because they have cell wall. So what will you do? This is where the knowledge that you have gained during cell, right, will come in handy. Remember Golgi body? So this Golgi body will synthesize vesicles containing calcium pectate and magnesium pectate. These vesicles will start aligning themselves at the center. Okay. So here you can see at the end of telophase, when the chromosomes have separated, these vesicles will start accumulating in the center of the cell. After accumulating, they will start fusing. Okay. And this will form the cell plate. It is a cell plate. It is nothing but fusion of such vesicles from Golgi. Along with the cell plate, there will also be some actin and myosin filaments, and this together it is known as phragmoblast. These vesicles fuse, and as the vesicles fuse, they are also made up of lipid membrane, right? So that lipid membrane will fuse with the cell membrane, and whatever its content was there, which was calcium pectate and magnesium pectate, will be deposited outside the cell. Fine. It will form what is called the middle lamella. And after middle lamella, you now have the cell membrane. And if the cell wants, this is the cell which has divided. And now if it wants, it will start synthesizing the primary cell wall. And this is how a plant cell divides. Okay, you can see the vesicles are fusing. This is forming of cell plate. Okay, and then later on, as it keeps on fusing and the calcium pectate gets deposited in between, the two cells will divide. This is the real life what you can say time lapse of the same thing. You can see here also how the single cells, they are dividing by formation of cell plate. Fine. Here also you will see the cell plate is forming, and then the cell itself will divide into two parts. Notice? Okay. In some organism, karyokinesis is not followed by cytokinesis. So there will be division of the nucleus, but there will be no division of the cytoplasm. In such case, there will be multinucleate condition formed. This is called syncytium in case of animals, and it is called choanocyte cell in case of plants. Remember? Example is liquid endosperm inside a coconut. Inside a coconut, there will be karyokinesis, which is division of the nucleus first. So you have a lot of nucleus in the cytoplasm. The coconut water that you drink is nothing but the cytoplasm of the cells, and there will be a lot of nuclei floating in that cytoplasm. Fine. What is the significance of mitosis? It keeps the nucleocytoplasm ratio, which means that if there is a nucleus, it has a certain area which it can control, right? So this area or this volume is controlled by mitosis. If the cell grows more than what the nucleus can control, it will undergo division. For development and growth of an organism, right from zygote to a well adult organism. Then for asexual reproduction of unicellular organism, which is called binary fission also, right? Or amitosis. Then cell repair, when there is any damage to the epidermis or any damage to other cells, right? And as the cells are dying, new cells are getting replaced. So for this cell repair. And finally, mitotic division in the meristematic tissue. Okay, it could be apical or lateral cambium. It results in continuous growth of plants throughout their life. So this is the significance of mitosis. Next time when we discuss, we will be talking about meiosis. Okay, see you next time.